Life process can be defined as a physiometabolic process that sustains life. It includes various physiological processes like nutrition, respiration, transportation, and excretion. Each physiological process is actually runs under the integrated system of chemical reactions, which is called as metabolism. So, if we see life, we find the different physiological process at the end of each physiological process. There is a metabolism works through a system which are correlated and interdependent, and in totality they appear as a life activities. So such process which are inevitable to sustain the life processes is called as life processes. Nutrition is a process of taking nutrient. So process of nutrition can be divided into two parts: autotrophic mode of nutrition and heterotrophic mode of nutrition. In autotrophic mode of nutrition, organisms derive food from natural resources, and in that case they are not depending on any other organism. As an example is green plants. They utilizes the light energy, carbon dioxide from the air, water from the soil, and they synthesizes a compound called starch, which is a type of carbohydrate. So in this case we see that for preparing the food they are no way dependent on any other kind of organism. If we look at the entire process of photosynthesis, we will see that 6 molecules of carbon dioxide is required along with the 12 molecules of water in presence of light chlorophyll pigment to provide 1 molecule of starch and 6 molecules of oxygen. In the process of photosynthesis, we see that the products can be categorized in two parts. First is the carbohydrate, which is the main part, main product, whereas oxygen evolved during the process is been considered as byproduct of the photosynthesis reaction. This is the transverse section of leaf. We can see in the diagram the leaf is been covered by two limiting layers called as epidermis. The epidermal layer which is found inside is called lower epidermis. We can see some pores in the epidermal layer area. These tiny pores are called stoma, and these are not the passive pores. Actually, they are being guarded by a bean-shaped cells that we can see in the figure is called guard cell. As we have seen in the previous slide, that the process of photosynthesis requires carbon dioxide, and plants evades carbon dioxide from the atmosphere. So stomata in this process help them to get it from there, and at the same time the oxygen which has been formed as a byproduct of the process also goes out through these pores. So stomatal openings help in the exchange of gases. Again, we can see in between the upper and lower epidermis there are two different types of cells inside: palisade and spongy cell. Palisades are the long elongated cell having numerous chloroplasts inside. They are the core area where actually the most of the photosynthesis process are taking place, whereas the storage cells having very fewer chloroplasts in them, and they stores the photosynthetes, that is the starch, once they are been formed. In the next slide, we are going to learn about the process of photosynthesis in chloroplast, because it is the chloroplast where all the type of chemical reactions required for the process of the photosynthesis is taking place. Now, let us see the process of photosynthesis taking place inside the chloroplast. As we have learned in the previous class about the chloroplast, it is a type of plastid, an organelle which help in the process of the photosynthesis. If we want to understand the chloroplast structurally, we will find it as a double membrane layer, and inside the inner membrane there is a sap which is called stroma. We also find stacks of membrane inside the chloroplast which is called as granum, and each granum is in turn having a smaller unit called as thylakoid. If we elaborate the process of the photosynthesis, we can divide it in two basic parts. First is the light reaction, that is the light dependent part. These membranous structure inside the chloroplast, which is called as granum, having many pigment system inside along with the chlorophyll. So these pigments actually traps, imbibes the radiant energy of the sun and help in the transmission of electron through a defined pathway that is called electron transport chain. But to make the process of flow of electron through the electron transport chain in continue, they require to have also the continuous supply of electrons. When the chlorophyll pigments traps the radiant energy, their electrons get excited and jumps to the higher orbitals, that in turn is being trapped by some other pigment molecules or electron acceptors which are found along the line of electron transport chain. But ultimately, these electrons are being captured by a coenzyme that has been shown in the figure, NADPH plus, showing there is a deficiency of electron in them. So NADPH plus coenzyme actually traps the oxygen and traps the electron finally. So who makes the continuous supply of electron throughout the chain? It is actually the water. These water molecules splits up by utilizing the sunlight and some cofactor like manganese and enzymes into protons and electrons. Protons are being excited by these coenzyme NAD, whereas electrons are passed through the electron transport chain. Actually, first they are being trapped by the chlorophyll pigments of photosystems, and thereafter they are being transferred to the next electron acceptor in the electron transport chain. So we can see that the flow of electron in the light reaction part of the photosynthesis is being actually maintained by the splitting of water molecule. The splitting of water molecule by utilizing the sunlight, and also this process has been assisted by certain enzymes, is called as photolysis of water. Finally, these NADPH molecules help in accumulation of the hydrogen atoms and generation of the ATP molecules. We can see in the figure that one ADP molecule combines with one PI, that is the inorganic phosphate, and forms the ATP. So what happens in light reaction part? NADPH and ATP is being formed. NADPH and ATP collectively called as assimilative power of the cell, as they help in the process of assimilation, that is the photosynthesis process. So this is one of the part of the photosynthesis that is taking place in the chloroplast, and that part is the light reaction. Once this assimilative power is been formed, now chloroplast is ready for another kind of reaction that takes place in its liquid part, sap part, that is the stroma. And it is not a pathway, but it is actually a cycle, and it is called as Calvin cycle in the honor of the name of its discoverer, Calvin Benson cycle. In this process, carbon dioxide is actually been reduced by the coenzyme NADPH, and carbohydrate is thus formed in the process. As a result, also 6 molecule of oxygen is released as a byproduct. So this is the overall process of the photosynthesis that we will also cover in the next slide, the basic 3 process in the process of the photosynthesis. So we have seen that chloroplast is the vital part in the entire process of photosynthesis. Entire process of photosynthesis is actually taking place inside the chloroplast. As we have seen in the previous slide that the chloroplast plays a very important role in the entire process of the photosynthesis. We have learned that the entire process of the photosynthesis can be divided into basic parts, that is light dependent part and light independent part. So light dependent part is actually called as light reaction part, and light independent part is called as dark reaction part. Light dependent part is called light reaction, and it is taking place in the lamellar system, that is the grana or thylakoidal system of the chloroplast. While the dark reaction, that is the light independent part, which is also called as the Calvin-Benson cycle, is taking place in stroma. So if we sum up the entire process of photosynthesis, we can easily make three major events in the entire process of photosynthesis. The event number first is the absorption of light energy by the pigments, basically the chlorophyll, and the next event, which is following the first, is the splitting of water molecules into hydrogen, oxygen and electrons. Here hydrogen is actually represented in the form of the proton. So this is also called as photolysis of water. So we can understand the oxygen which comes as a byproduct at the end of the reaction is being actually sourced by water as a result of process of photosynthesis. So at the end of the reaction, if we can recall the previous slide, we have learned that light reaction ends into ATP molecules and NADPH, that is coenzyme which contains hydrogen ions, and these hydrogen ions was actually formed after the splitting of water molecules. So these togetherly called as assimilative power of the cell. Both are required in the next part of the reaction, which is called dark reaction. So dark reaction is taking place in the stroma, where carbon dioxide is being accepted by a compound, and this compound is being assisted by an enzyme which is called as Rubisco, ribulose biphosphate synthetase carboxylase oxidase. So this Rubisco enzyme actually help in the fixation of carbon dioxide in the chloroplast. Further, this carbon dioxide molecule is being reduced by the NADH. That NADH, which was the product of light reaction part. Also, the dark reaction requires energy, and we have seen the light reaction part has also yielded ATP molecules. So light reaction part we can understand as energy generator, which evolves ATP molecules and the reducing factor NADH, and that reducing factor is actually utilized in the dark reaction part to reduce the carbon dioxide into the carbohydrate molecule. So at the end of the reaction, we find carbohydrate in the form of starch is been formed, and this process also yields oxygen as a byproduct. So this way the entire process of photosynthesis has been accomplished inside the chloroplast. In the next following slides, we are going to learn about few activities, basically two activities. One activity is there to understand how chlorophyll is important in the process of photosynthesis, and another activity is about the requirement of carbon dioxide for the process of photosynthesis. If a cross section of leaf is seen under a microscope, we can see many green dots in them. These dots are actually the chlorophyll pigments. Chlorophyll is a very important compound found in chloroplast, helps in trapping the sunlight and providing the energy to convert carbon dioxide into the starch. All these things we have learned in the previous slides. So to see that chlorophyll is playing a very important role in the process of photosynthesis, we can have an activity. To perform this activity, we take a variegated leaf. A variegated leaf means a leaf with multicolor lamina. We will see that it has red, blue, green, yellow, orange, pink, many colored patches are there. So first of all, we will take a variegated leaf and then we will mark its different color patches so that once the leaf is been decolorized, we will able to identify which area belongs to what kind of color patch. To decolorize the leaf, we use alcohol, as alcohol is a organic solvent and chlorophyll is a organic compound. So when we boil the leaf in alcohol, after some time the leaf becomes decolorized due to dissolution of these pigment in alcohol. So once the leaf becomes decolorized, we take it out from the alcohol, wash it with water, and then dry it with the help of a filter paper. Once the leaf has been dried, we will identify with the help of the marking that we have done before boiling the leaf, decolorizing the leaf, and put the iodine solution on each colored area. We will see that the areas which are non-green in nature never shows positive iodine test. That means they doesn't become blue. But once we put the iodine solution on green patches that we can see in the diagram, it turns blue. We have learned in the previous classes that iodine, when reacts with the starch, changes its color from pink to the blue. So iodine is taken as an indicator to assess whether the starch is present or not. So what is the assertion of this activity? We have seen that it is the chlorophyll pigment that helped in the process of the photosynthesis. How? Because we know that in the process of the photosynthesis, at the end, we get starch as a main product. So if starch is there in the green part, so it confirms that chlorophyll is one of the most important component for the process of photosynthesis. Similarly, we can perform another activity to show presence of carbon dioxide essential for photosynthesis. To perform this activity, we'll have to take two potted plants. So there will be requirement of two potted plants, and both of the potted plants, as you can see in the figures 5a and 5b, are being kept inside a bale jar. The only difference in both of the potted plants is the presence of potassium hydroxide, which we see in figure A but not in figure B. So before putting the bale jar over the potted plants, we will make it correct that no air is coming inside the bale jar from outside, and similarly air is not getting outside of the bale jar. For that, we will put Vaseline at the end of the bale jars. Now both of the plants will be left for 24 hours in sunlight, and after that we will pluck their single leaf, and we will repeat the same process of decolorization. That is, we will boil both of the leaf in an alcohol solution, and once they have been decolorized, we will perform iodine test on them. We will see when the iodine test is being performed on the leaves of both of the plants, the leaf from the plant A that we can see in the figure, where there is a petri dish with potassium hydroxide, doesn't respond positively, whereas the another potted plants in figure B that we can see, where KOH is not there, it responds positively toward the iodine test. That means its leaf turns blue. So how did it happen? So KOH is being a CO2 absorbent, has absorbed entire carbon dioxide in the atmosphere of the potted plant A, locked inside the bell jar. While in the potted plant, which has been seen in the figure B, as it doesn't have the KOH, so had the carbon dioxide available in its environment. Therefore, it successfully done the process of photosynthesis. That is why we can see the presence of starch in their leaves. So, from this experiment, we can again reconfirm that carbon dioxide is one of the components required for the process of photosynthesis. Now let us try to understand the stomata. As we have seen in the previous slide, that stomata are the tiny pores formed as a result of modification of epidermal cells. They are not the passive pores, they are active pores. Active pores means they remain closed during the night time but open during the daytime in normal circumstances. These are a very important structure in plant leaf. If we analyze their guard cell, we will see that the guard cells are the reniform or bean-shaped structures having thickened inner wall, and it contains numerous chloroplasts inside with nucleus. The surrounding cells of the guard cells are called as subsidiary cells. They are also the modification of epidermal cells. Stoma does a very important role in the plant, as we have learned in the previous slides. They facilitate the exchange of gases, and at the same time they also help in the loss of water in the form of vapor, that is called transpiration. However, the older parts of stem and roots are having some passive opening. That means they don't have any guard cells. They remain open during day and night. They are called as lenticles. So we have seen there are certain pores in the leaves called as stomata, that the number of stomata when we see or we compare from upper and lower epidermis, we find that the number of stomata in the lower epidermis is always higher than the upper epidermis. It is in order to check the loss of water through the process of transpiration. If there is more stomata are present in the upper epidermis, then the loss of the water will be higher and the plant will die of desiccation. Stomata are the pores on the leaf that we have learned in the previous slides. These pores are being protected by means of guard cells. We can see the structure of guard cell in the slide. They are reniform. Here in this slide we can see that stomata having two types of positions. One is the open position and other one is the closed position. During the daytime, photosynthesis is taking place in guard cells because the guard cells are having a numerous chloroplast in them. So with that they are making starch, and later on these starch are being converted into the sucrose, which causes hypertonic condition in the guard cell, leading thereby the endosmosis inside the guard cells. Due to endosmosis, the pressure is exerted on the lateral walls of guard cells because the inner walls are very thick in comparison to the outer walls. As a result of that, the guard cells opens the pore of the stomata. But on the other hand, during the night, when there is no photosynthesis, the guard cells not making the starch and the sugar, and the process has been reversed. Therefore, due to the exosmosis, water comes out to the surrounding cells, subsidiary cells and other epidermal cells, and stomata get closed. So this is the mechanism by which stomata opens and closed during the day and night time under a normal circumstances. Finally, we will see, apart from the photosynthesis process, plants also performs other activity of absorption by which other nutrients other than the carbohydrates are being formed. In the next slide, autotrophs, apart from the photosynthesis process, in that it makes carbohydrate, but apart from the carbohydrate, plants have several other requirements of chemical metabolites like proteins, enzymes, other complex organic molecules, fats. So therefore, the other chemical compounds are being formed by utilizing the energy of this photosynthesis, that was the output of the photosynthesis process. Like we can understand it by the example of nitrogen. Nitrogen is being absorbed by the plants through their roots, and further in their cells they make amino acids and protein finally. Similarly, other nutrients are also being utilized by the plants in their cells for making the different kinds of chemical compounds. So we can understand that the carbohydrate which was formed as a result of the photosynthesis output has two fates. First, it is utilized for the energy purpose, and second, it may be one of the kind of the different chemical compound that remains stored in the plant body as a biomass. Thank you for watching the video.